Chapter 1, Introduction to Microbes and Their Building Blocks. So micro, that word part means small. You've probably heard a lot of words that have that in it, and microscope and whatnot means small. The word microorganism and the word microbe mean the same thing, so if we use those interchangeably throughout this course, make sure you don't get mixed up about that. So microbiology deals with things too small to be seen without magnification. So we use things like microscopes, and there are various types of microscopes that we'll talk about. We'll mainly be concerned with one called a light microscope. So the microorganisms we're going to talk about this term are bacteria, algae, protozoa, and these worms called helminths. So microbes are very easy to study in some regards because they reproduce rapidly, and so we can grow a lot of them in the lab very, very quickly. On the other hand, though, they are very difficult to study because they can't be seen directly, and so we have to use things like microscopes to be able to see them. And some of the things on them we can't see at all. So which of these is not considered a microbe? Bacteria, algae, mushroom, or a protozoan? You probably already know the answer, and of course it is mushroom. So some sort of a bacterial type organism is estimated to have been on the Earth for 3.5 billion years. So for a long time, they were the only things on the Earth, about the first 2 billion of those years. And those beginning bacterial type organisms, those microbes, were called prokaryotes. And they had no true nucleus, so they called a prenucleus. So they have some of the material that we find in a nucleus, but they weren't wrapped up in a nuclear membrane that we'll discuss later. Eukaryotic organisms, or eukaryotes, we started to see on the Earth about 1.8 billion years ago. Eukary means true nucleus, and so these began having a nuclear envelope around the nuclear material. So they are the precursors to the organisms to come later, which were multicellular organisms like people and dogs and whatnot. So microbes are said to be ubiquitous, which is a fancy word meaning that they're all over the place. There's pretty much nowhere on the Earth that you can't find them besides maybe inside of lava or something like that. Okay, so we have them way deep in the Earth's crust. We'll find them in the polar ice caps, deep in the oceans, even as deep as thousands of feet down, and in temperatures of hundreds of degrees around the vents in the bottom of the ocean. We find them inside of the bodies of plants, animals, including humans, as well as insects like termites and things like that. So they occur in large numbers. They live in places where other organisms can't survive. They've been on the Earth for so long that they've been able to, over time, adapt to extreme environments that other organisms have not. Okay, so they serve many roles essential for life in other organisms. I gave a couple of examples. In humans, we have what we call the normal flora, which aids in things like digesting our food, or simply competing with pathogenic organisms like fungi and bacteria and whatnot. In termites, I gave that example, and ruminants like cattle, they don't have the ability to digest the cell walls of plants, and so they have bacteria living in their gut that actually breaks the bonds between the glucose molecules and the cellulose so that they can actually digest them. Okay, so they call those symbiotic relationships. So this just shows a evolutionary timeline of when we began to start seeing these things. So 4 billion years ago is somewhere around the estimate of when the Earth began. prokaryotes we started seeing somewhere toward the 4 billion mark, eukaryotes, insects, reptiles, mammals, and pay attention to humans here. In this whole evolutionary scheme, we're one black line. Okay, so not very much. So let's talk about evolution for a little bit. I don't think we need to get into a religious discussion about this. So evolution is the accumulation of changes that occur in a group of organisms as they adapt to their environments. So the theory of evolution is what it's called, but it is well documented. It's observable and testable. We can prove that evolution occurs by doing experiments because the controlled experiments that we can do short term are the basis for evolution over long term. And so if you can accept that, you know, a group of animals, like a group of wild horses or wild dogs, can change in a short period of time because of changes in their environment, all you need to do is extrapolate that data to a long period of time, and that is essentially what evolution is. Never been disproven. People have been testing it for years, and yes, there are people who definitely want to disprove it, but so far it has not been able to be disproven. There are definitely some specific theories of evolution that have been disproven over time, but the overall and mostly current understanding of it is fairly well accepted. Okay, so how microbes shape our planet. Now, there's this thing called photosynthesis, and microbes are able, along with other organisms, to convert organic molecules into other molecules using the sun's energy, such as taking carbon dioxide and water and turning it into sugar. So anoxygenic photosynthesis is the light-fueled process of the conversion of CO2 to organic material without using oxygen. Okay? So this is how they think that it began, and then over time they think that it led to oxygenic photosynthesis, which of course is photosynthesis that produces oxygen. This is how we get oxygen on the earth today. So plants and algae and certain bacteria will take things like carbon dioxide and use it, and the waste product that they give off, thankfully, is oxygen. So oxygenic photosynthesis is the light-fueled process of converting carbon dioxide into organic material. Like I said, the example was sugar, such as glucose or cellulose. And as I said, it produces oxygen, and this is the source of the oxygen that we have on the earth. So it led to the use of oxygen for aerobic respiration in other organisms like bacteria or animals. We all use aerobic respiration, which means we have to have oxygen in order to create ATP using glucose. So photosynthetic microorganisms account for about 70% of the Earth's photosynthesis. The rest comes from multicellular organisms, which are trees and plants and those sorts of things. So microbes are the main forces that drive the structure and content of the soil, water, and atmosphere, meaning they are a major part of the composition of all of those things. They produce gases like carbon dioxide, nitric oxide, methane, which is what natural gas is. They help regulate the temperature of the earth. Bacteria actually do create some heat. If you've ever looked at a compost pile or a silage pit on a farm, you can actually see steam coming off of them in the winter because of the bacteria and other microorganisms inside of it that are giving off heat. Okay, so there's a huge underground community of microbes that influence weather. They can use them for mineral extraction, which I have no idea how they do that. But as I said before, they're also used for soil formation, composting organic products, turning it into things that we call fertilizer that can be used by plants to grow. So bacteria live in close association with other organisms. I mentioned that a little bit earlier, where termites in the guts of ruminants, but also there's a lichen that has a bacteria that lives in it, and I mentioned humans and probiotics. So it's estimated that microbes that live under the Earth's surface account for 50% of the total microbes. So true or false? The theory of evolution is not widely accepted among scientists because it still remains unproven. And of course the answer is true. So let's talk about the interaction of microbes and humans for a while. Okay, so the historical uses of microbes by human include things like bread production, because we use yeast, which is a fungus, to make bread rise and things like that. We use it in alcohol production, also yeast, things like beer. Thank goodness, because we use the yeast to digest sugars such as maltose inside of the grain, and one of the byproducts that they give off is alcohol. It's used in cheese production. So bacterial and fungal cultures are used to produce cheeses and yogurts and things like that. It's also been used for the treatment of wounds and lesions, and is still today, because penicillin, which was arguably the greatest breakthrough in medical history, was discovered because of mold on a piece of bread, and noticing that the bread didn't spoil or something like that. Somehow it's used in mining precious metals. I have no idea how. Cleaning up human-created contamination, sewage treatment, for example, oil spills. In the Gulf oil spill, the BP oil spill, they used bacteria, and I even not used, but just naturally, the bacteria began to digest the oil spill. It's used in genetic engineering, okay, something called recombinant DNA technology. We use it for insulin production, hybridization, so we can hybridize different organisms. But it's this area of biotechnology that takes genetic material from one organism to another organism, what we call genetically modified organisms, or GMOs, which is a huge controversy in food nowadays. So recombinant DNA technology is the transfer of genetic material, like I said, from one organism to another. There's something called bioremediation, which is what I mentioned earlier, using microorganisms to clean up toxic pollutants or things like that. So the majority of microorganisms that are associated with us cause us no harm. To the contrary, they're actually quite necessary for us to function properly. However, there are these things called pathogens, and those are microbes that actually cause disease. So the World Health Organization estimates that there are 10 billion new infections caused every year by microbes. Things like tuberculosis, chickenpox, the common flu, the dreaded bird flu, and so on and so forth. So infectious diseases are among the most common causes of death in the world and the nation. So about 13 million people die worldwide per year. So it's estimated that a child dies from malaria, for example, every 30 seconds. Okay, so there are some new diseases, new relative to the age of the earth, I suppose. In my lifetime, I can remember when AIDS began, hepatitis C, viral encephalitis, all fairly recent diseases. However, there are also diseases that have been around for centuries that we are beginning to see coming back, such as smallpox, tuberculosis, measles, things like that. And interestingly, some of the diseases that used to be considered non-infectious, meaning that they didn't come from a microbe, are now being shown to have a pathogenic microbial cause, such as gastric ulcers. H. pylori was discovered, I think, by an Australian man within the last 20 years to cause stomach ulcers. Diabetes has been linked to something called the Coxsackie virus. Schizophrenia by something called the Borna agent. And they've been able to link chronic diseases such as MS, OCD, coronary artery disease, obesity to chronic infections of bacteria and viruses. So this is a table that just shows the top causes of death, and you can see that number 4 is respiratory infection. Number 7 is AIDS. This is worldwide. Number 9, diarrhea. That would be things like malaria. Number 10 is tuberculosis. If you look at the United States, it's quite different. But number 8 and 10 are infectious diseases. True or false? Diseases such as heart disease, gastric ulcers, diabetes, multiple sclerosis can have a microbial cause. And of course, the answer is true. All right, let's discuss some of the historical aspects of microbiology. So there's this thing called spontaneous generation, and it's a concept that there was some sort of vital forces present in different forms, different types of matter, like, peanut butter or a piece of bread or something like that, that led to the creation of life. And it basically stemmed from the fact that when food and whatnot was left out, things would begin to grow on them. Okay? And so eventually they discovered microbes, but this theory of abiogenesis, a meaning without, bio meaning life, genesis meaning to generate, so generation of life, or generation without life, I guess is what that literally means, continued on even after they discovered that it was actually because of microorganisms that this stuff began to grow. So of course others advocated what's called biogenesis, which is what we believe now, is that life comes from other life. So let's talk about some people in microbiology history. Louis Pasteur studied the roles of microscopes in fermentation of alcoholic beverages. He also used a series of experiments with these things called swan-necked flasks, which look like this, to be able to disprove abiogenesis. So what he did was he filled these flasks with a broth, something that microorganisms he knew that they could grow in, and had these long openings on them, this stem on the top of them that he called a swan-necked flask, of course, because they look like a swan neck, right? Then he heated them up to sterilize them. What that does is kill all the bacteria in them. So some of the flasks he exposed to dust. The ones that had the swan necks on them, of course, didn't, because if you look at this picture here, microorganisms and dust can't get in there because they have no way of crawling up this tube. So they could get only to this very bottom part right here. And so the experiment, of course, showed that there was no such thing as abiogenesis. And so this picture in your book shows how if the neck of the stem is broken or short, or of the flask, then the microbes can get in. If it is not, they have no way of getting in, and so this broth over in the swan neck flask remains sterile. Okay, so the most important thing regarding microbiology is the invention of the microscope. So this person named Robert Hooke began building these rudimentary microscope type things to be able to study all kinds of things, plants, trees. Leeuwenhoek took the idea of the magnifying glass and began manufacturing slightly more complex things that eventually led to the microscope. Okay, so he would look at threads on fabrics and things like that. And he was looking in drops of rainwater and saw little things swimming around here, in them, that he called animals. He also called these things animalcules, which is sort of a combination of the term molecule and animal, sort of put together, that he scraped off of people's teeth. So there are a few major advancements in microbiology that we're going to talk about. There are these things called restriction enzymes that were discovered in the 70s by these three scientists. And what they are basically these things they call molecular scissors that are inside of prokaryotic organisms. What they do is they split up or break up the DNA into smaller pieces. And so what they've done is they've been able to take those scissors and cut up DNA in the lab, and that's what allows us to splice DNA and create genetically modified organisms. In the 80s, the invention of the polymerase chain reaction technique was invented. And what this does is allows us to take a little bit of DNA, and the word they use is amplify, but it's really multiply it, because you're actually taking that DNA and you have more building blocks of DNA, and we basically replicate the DNA into huge quantities to allow us for studying. You've probably seen those pictures of those lines. It looks like a bar graph with little tiny lines going up and down it. Maybe on crime scene shows like CSI or something like that, and that's what you're looking at is this DNA that's been amplified, and then if they run it through something called a gel electrophoresis to actually pull it apart. We'll probably get to that sometime later in the book. So what this has done is given us the ability to be able to understand and tell the difference between microorganisms that otherwise look very, very similar. It's used for studying and diagnosis of infectious diseases because they can look at the nuclear material of the microorganisms and determine what the microorganism is. Forensic work, I mentioned crime scene investigation. So what they'll do is if they find a piece of DNA on hair or clothing or some sort of a semen sample or something like that, they can then match that DNA using this polymerase chain reaction to the suspect. So a man named Kary Mullis is the actual inventor of this, and he won the Nobel Prize for Chemistry back in 1993, which incidentally is the year I graduated high school. So this shows a picture of the polymerase chain reaction. We take one strand of DNA, and it's a exponential growth. Okay, so 1 goes to 2, 2 goes to 4, 4 goes to 8, 16, 32, 64, 128, 256, and you get the idea. 612, 1028. It's, if you didn't notice that, those are all the same numbers, by the way, that they use in computers for things like RAM and whatnot, because they double the same way. Okay, in the 80s, 90s, and 2000s, they began discovering these things that they call biofilms that they are noticing were present in certain types of infectious diseases. And so what it is these accumulations of bacteria and other microbes in some sort of a film that were associated with these different types of diseases. So often they find multiple species in one biofilm, and they may find biofilms on top of biofilms on top of biofilms. So they've known this in microbiology for a very long time as far as environmental goes, but now they're starting, well, and by the way, this is in ship hulls. They've seen it on ancient paintings. They see it on rocks and so on. But now what they're noticing is that they're finding these things in the human associated with some sort of a disease process, like dental plaque is a biofilm, certain ear infections, prostate infection, and so on. So these are a particular problem for implanting things into the body, such as artificial hips, hearts, valves, IUDs, which are birth control, you probably know. So anyway, it can cause those things to not be a success. So in the 2000s, they began to start to understand better these things called small RNAs. Okay, so previously they thought that DNA made RNA, which led to the creation of proteins, and that is true. DNA does lead to RNA because you get RNA from the DNA, and RNA. By the way, I guess I should tell you, RNA stands for ribonucleic acid. It's a single strand of nucleotides. DNA stands for deoxyribonucleic acid, which is a double strand of nucleotides. We'll talk about that in detail a little bit later. But the RNA, there's different types of RNA that we'll talk about. Some of them become part of this structure, which is this blue thing that you're looking at here called a ribosome. That's actually not true. That's showing a picture of the silencing complex. So I apologize because it looks exactly like that. Anyway, it becomes part of a ribosome, which is the structure that actually makes the protein inside the cell, either on the rough endoplasmic reticulum or just floating around in the cytoplasm. But what we're talking about here is that these small RNAs are found to not be used for making a protein. Okay, so they think that about only 2% of the DNA leads to making a protein. Okay, now it needs to be said, especially in higher animals like humans, that a lot of the DNA is not used at all for anything because we have genes that are suppressed from an evolutionary standpoint, such as the genetic material to build a tail. We don't use the genetic material that is the instructions for having long body hair like an ape or a monkey. We have it, but we don't use it, and so on. Okay, but what these small RNAs are small chains of RNA, and they don't really understand exactly what it has or what it does, but they think that it has some role in regulating what happens inside of the cell. Okay, now you should understand we don't know very much about how, well, I should say we know a lot, but there's a lot that we don't know about how the body works, or for that matter, other organisms. But what they're using this for is to be able to treat diseases because they can do things like disrupt this process of creating these small RNAs in pathogens to be able to kill them. Okay, so other people important in the history of microbiology. This person named Robert Koch. He developed these things that we call Koch postulates, okay, to be able to verify that diseases were actually caused by germs. So he established a link between a microbe and the disease it caused. He actually discovered that anthrax was caused by the bacteria Bacillus anthracis. We still use his postulates today for discovering diseases. These people, Holmes and Semmelweis, they discovered the importance of what we call today the aseptic techniques, which is A means without, sept means bacteria, and so techniques to prevent the spread of bacteria, things like washing hands and sterilizing chemicals and those sorts of things. And what they figured out was they noticed that people who had home births resulted in fewer infections than the people who had babies at the hospital, and what they discovered was the doctors were leaving the morgue and coming over and delivering babies and not washing their hands because they just didn't understand that that's where the diseases were coming from. Lister began to help develop these aseptic techniques in surgery in the 1800s, and a person named Cowan discovered these things called endospores, which we'll get to in a later chapter. Okay, so who's the scientist who built the first simple microscope? And of course it was Leeuwenhoek. Okay, so there's two types of cells that we're going to discuss. Really, there's only two types there are as far as cells goes. This is the first category when we're dividing up phylogenetically organisms. So the first one are called prokaryotes. The other one's called eukaryotes. So prokaryotes, 10 times smaller than eukaryotes. They do not have organelles, and all prokaryotes are microorganisms. There are no multicellular prokaryotes. Okay? Eukaryotes do contain organelles, which are the literally means little organ. Okay? So these are these small double membrane bound structures, all that we find inside the cell, like mitochondria, endoplasmic reticulum, Golgi apparatus, things like that. Chloroplasts in photosynthetic organisms, the nucleus, and so on. Okay, so only some of the eukaryotes are microorganisms. Okay, the first ones were microorganisms, single-celled organisms. However, they eventually evolved into multicellular organisms, and so all the multicellular organisms that we have on the planet today are eukaryotes. So they have membrane-bound nuclei and organelles and so on. Of course, you can see macroorganisms or multicellular organisms without the naked eye, or without a microscope. Okay, so viruses. These are not independently living organisms. So it's arguable whether they're even alive at all. Not everybody agrees on that. They have to have a host, some sort of an animal to live in, because when the animal's cells that they are infected with, let me start that over. The animal cells that the viruses are infected in, when they replicate, it replicates the virus with them, and that's how the viruses reproduce. Okay, so they're basically just hereditary material, either DNA or RNA or both, depending on the virus, and some sort of a protein coat. And they have some fairly elaborate structures that we'll get to later on. So when they're inside the host organism, they exist as just genetic material. And what they do is they splice themselves into the DNA of the host organism. Okay, so this is just a picture of some examples of the different types of organisms that we're going to talk about. Okay, a helminth. This is a Tinea solium, the herpes simplex virus, which causes cold sores. A protozoan, E. coli, Escherichia coli, which is what lives in the colon of many animals, and this type of fungus. There's all different kinds. This is just an example. And by the way, this is showing it in comparison to a human hair to give you an idea of the size of these things. Oh, and I should also note, there are various sizes of these things, so they're not all, not all bacteria are this size, or for that matter, they're not all that shape. Interestingly, this is the size of a red blood cell right here. Very, very small. Okay, so place the following organisms in order from largest to smallest: protozoan, virus, bacterium, and helminth. And the order is helminth, the biggest, then protozoan, then the bacterium, and then a virus. Viruses are very, very small. Okay, everybody's favorite subject, chemistry. So we're not going to go into too much detail. You should have had chemistry in at least one other class prior to this, probably a couple or several even. But it's worth a little bit of a review. Okay, so macromolecules, so larger molecules, the structures of life. So very large molecules, there's four main categories of macromolecules. Carbohydrates, people call them carbs, they're sugars. Lipids, which are fats, proteins, which everybody knows what those are, and nucleic acids. Okay, so there's this word called monomers. Mono means one, right? So the monomers are the subunits of these macromolecules. So polymers, poly means many, would be chains of monomers. So you put monomers together, you end up with polymers. Okay, now this is a table in your book. You can use this to refer back to it. We're not going to go over anything right now, but make sure that you go back and look this over when we're finished with this. Okay, and this also shows some pictures with some narration on the different types of molecules that we're going to talk about. Okay, so carbohydrates, generally represented by the chemical formula CH2ON to the how many. Okay, and what that means is for every carbon, you're going to see two hydrogens and an oxygen. Okay, so for example, with the molecule of glucose, it has 6 carbons, and so the chemical formula is C6H12O6. So you have the same number of oxygens as you have carbons. So monosaccharides and disaccharides, mono means one, and saccharide means sugar, di means two, are specified by combining a prefix that describes some characteristics of the sugar with some suffix, or with the suffix "-ose". Okay, for example, hexose. Hex means 6, therefore a hexose sugar has 6 carbons. Pentose, pent means 5, like a pentagram, and so it has 5 carbons in it. Okay, so let's talk about some specific monosaccharides. Glucose. Now, glucose is sweet, and it is the most common hexose. It is the sugar that humans use to convert into energy so that we can create an ATP. Glucan, by the way, means sweet as well. Fructose is a fruit sugar. Oh, and I should say glucose is the type of sugar we have that is regulated in our blood. Fructose is fruit sugar, not only found in fruit, but it is certainly in fruit, and that's where it gets its name. Galactose is what we call milk sugar, and again, these are all monosaccharides, meaning only one molecule. Okay. Some disaccharides are lactose, which is milk sugar. It's a disaccharide of galactose and glucose. Maltose is malt sugar. We get it out of wheat. Sucrose is cane sugar or table sugar. So this is a fructose and a glucose. Oh, and by the way, maltose is 2 glucoses. Okay, so let's talk about some polysaccharides. So polysaccharides are used for structural support and protection, as well as nutrient and energy stores, such as glycogen and starch. Cellulose is one that is found in the cell walls of plants and algae. So cellulose is wood, literally. And you may be surprised to know that cellulose is sugar, but wood is basically pure sugar. It just happens to be a type of sugar that we don't have an enzyme to break apart, and so we can't get any kind of food value out of it. But it's also seen in algae as well. Agar is something we're going to talk about a lot, but it's a polysaccharide that we use to culture microbes on. We put it in petri dishes and in test tubes and things like that. Peptidoglycan, polysaccharides or glycans linked to peptide fragments. Peptides are little chains of amino acids. And so we find these peptidoglycans on bacterial cell walls. Lipopolysaccharides are lipids with and polysaccharide complexes that are responsible for the symptoms of fever and shock. So they are released into the bloodstream and create those scenarios. A glycocalyx. Glycocalyxes, or calyces I should say, are on the outer surface of many cells. They function as attachments for receptors that receive external stimuli. For example, receptors for hormones on the cell surface. You'll also see these acting as the cell markers on the cells that we call antigens. Okay, so let's talk about lipids for a little bit. So there's 4 types of lipids: triglycerides, which we call true fats; phospholipids, which are a lipid with a phosphate group on it; steroids, which cholesterol is a steroid; steroid hormones like testosterone and estrogen are examples of steroids; and waxes. Okay, so let's talk about triglycerides for a little bit here. So this is how we store lipids, and this includes fats and oils. Oils you'll see in plants, fats, well, that's maybe a bad example, but this is what makes up our fat in people. So what a triglyceride is a glycerol molecule, which is this right here. This all in this pink right here. And 3 fatty acids. Okay, so now this isn't hooked to it yet, but this shows 3 fatty acids, and you probably heard that term fatty acid, like omega-3 fatty acid or omega-6 fatty acids. And so what it is a carbon chain with an oxygen and a bunch of hydrogens attached to it. Okay, so when we make a triglyceride, what we do is we attach this these 3 fatty acids to this glycerol molecule. Okay, so those carbons, the reaction we make to attach these is similar to a peptide bond, where we get rid of a water in order to attach these. So you can see we lose a hydrogen off of the glycerol and a hydroxide group off of the fatty acid, and we end up creating a water for every single one of these fatty acids. So fatty acids, as I said, are long chain hydrocarbon molecules. Saturated fatty acids, that means that all of the carbons have the maximum number of hydrogens bonded to them. Okay, so look right here. This is a saturated fat. Okay, saturated, the word saturated means that it's full, right? If you saturate a sponge, you soak up the sponge full of water or whatever, and it's full. If it's unsaturated, that means it's not full, and you can see that there are some hydrogens missing on this unsaturated fatty acid over here. Now, saturated fatty acids are usually solid at room temperature, not always. Usually from animals, not always. Coconut oil is an example of a saturated fatty acid, or a saturated fat that is not from an animal source. Saturated fats have been sort of vilified, for many, many years, because they've been implicated in heart disease and cardiovascular disease and whatnot. They're starting to realize now that that is not the case. Matter of fact, not only are saturated fats not really considered to be bad by a lot of people, they're actually considered, and there's a lot of research to back this up, that they're actually extremely necessary for optimum health. So unsaturated fatty acids, usually liquid at room temperature and usually from plants. So this would be things like canola oil, olive oil, those sorts of things. Okay, so in most cells, triglycerides are stored in long-term concentrated form as droplets or globules. So that's what you'll see inside of the fat cells of a human, for example. Okay, so membrane lipids. These are phospholipids. Now, membrane lipids, we're talking about the membranes of cells. So over here on the right, this is showing a cell membrane, just a little section of a cell membrane. And this is a phospholipid right here. Okay, so what's unique about these compared to a true fat, which are nonpolar, these phospholipids are polar on one end and nonpolar on the other end. Okay, and what that does, if a molecule is polar, it's able to create hydrogen bonds with water, okay, or other charged molecules or atoms. If it's nonpolar, it doesn't create any of those charging type bonds. And so the tail of a phospholipid is what we call hydrophobic. So phobia is a fear. Hydro means water, and that just means that it doesn't create bonds with water, because water is also a polar molecule. It has a positive end and a negative end, even though the global charge of the water molecule is neutral, because the nucleus of the oxygen hogs the electrons. It makes that end negative, where the end with the hydrogens attached to it are more positive, and so the negative end will be attracted to positive things and vice versa. Anyway, so because we put this phosphate group on here, it makes the phosphate end hydrophilic, so water-loving, and so it will create bonds with water. Okay, and so what you see is you get this arrangement called the phospholipid bilayer, where all of the tails are toward the inside because they don't want to be near the water, and all of the heads are on the outside. So their water will be on both sides of this membrane. And I pretty much explained all that. Okay, so these lipids naturally assume this single and double layers, these bilayers, which contribute to the significance in membranes. That's why they're used for membranes. Now, really quickly, you also see proteins, and this is the glycocalyx that we talked about earlier. Proteins, and these are actually cholesterol molecules embedded, which we're going to get to in just a minute. Okay, steroids and waxes. So this is actually showing this is a cholesterol molecule right here. So we got four rings, these carbon rings with this carbon-hydrogen tail hanging off the end of it. So steroids, found in cell membranes and also other animal hormones. Okay, cholesterol, which is a type of steroid, is a sterol, and so it's a steroid with a hydroxide group on it, right there. This is used to reinforce the cell membrane. What it does is it basically makes the cell membrane less fluid. Waxes, these are esters formed between long-chain alcohol and saturated fatty acids. They're non-polar, which means they're hydrophobic, and so various organisms like ducks will use these for waterproofing. That's when you see a duck preening, it's taking oil from its preening glands and spreading them all over its feathers, and that's how duck feathers are waterproof. Human skin, though, leaves, exoskeletons of insects, so we'll see this in various places, okay? Bacteria, the ones that cause tuberculosis and leprosy, also contain these waxes, or they create them, and that's one of the things that makes them hard to destroy. Proteins, okay, so this is the predominant organic molecule in cells. Okay, so they are polymers made up of monomers called amino acids. And over there on the right is a list of all of the amino acids. There's only 20 of them. You can refer back to this if you need to. I'm not going to make you learn every single amino acid. But they're the building blocks of proteins, 20 of them known, as I said. Okay, so let's discuss some of the terminology associated with proteins. So a peptide, this is a molecule composed of short chains of amino acids. Basically, a peptide, I mean, the nomenclature is not universal, okay, so people will use these in different ways. But in general, a peptide is considered to be a short protein, okay, but technically, a peptide is a protein. It's just amino acids put together. Now, you don't want to get that mixed up with a peptide bond, which is the bond that's formed between the amino acids when you remove a water molecule, meaning you remove a hydrogen and an OH group. A polypeptide is many peptides, right? Or many amino acids put together. There's no exact number here, but it's a lot usually, okay? So a protein is considered to be a polypeptide, but you can kind of use the word protein to sort of describe all of these. So if we use these kind of interchangeably, try to not get too mixed up. Okay, so proteins have 4 levels of folding that give them their structure. And one important thing about proteins to know is that structure determines function. Whatever shape the protein happens to be will determine what that protein does. Okay, so the first level is called the primary structure. And this is nothing more than the type and the number and the order of the amino acids in the chain, meaning which amino acids there are, how many of them there are, and the order. Now, the analogy I use for a couple of things, including this, is the analogy of the English language. Okay, so the equivalent to a protein would be like a word in our English language. Now, for our alphabet, we have 26 letters, but the alphabet of a protein has 20, right? The amino acids. Now, what determines what word we end up with are 2 things: the number of letters we use and the arrangement of the letters in the word, right? And so it's the same thing with a protein. The number of amino acids and the arrangement of the amino acids will determine what protein we end up with. Okay, so the primary structure is this right here, this line, and you can see this better in your book. But this is just a string of amino acids put together. Okay, now what happens is the primary structure can become a secondary structure. Okay, and what that is a folding. Okay, so we find these as functional groups on the outer surface that form hydrogen bonds with each other. So these groups on the surface of these amino acids will bond with other groups on other amino acids, and what that does is it creates various structures, such as an alpha helix or a beta pleated sheet, or both. Okay, so this right here is a beta pleated sheet. And so we form these bonds from these peaks to the other peaks, and it causes these pleats to be formed, so it's sort of corrugated. An alpha helix is means to twist, like a helicopter, and so this helix forms this helical structure, or this twisted structure right here. Now we may see, like I said, combinations of both. So a protein may have beta pleated sheets and alpha helices in the same protein. Okay, so we'll further fold into what we call tertiary structures. Okay, so these can be created by covalent bonds called disulfide bonds, which are covalent bonds between sulfurs on these functional groups, or other bonding covalent bonds, or for that matter, bonds between charged functional groups as well. But what it does is it gives us this sort of a structure here. Quaternary structure is formed when we take several, or more than one, I should say, polypeptide and put them together. So if you look down here, we see 3 different tertiary structures all bonded together into this is what we would call a globular protein. So each protein develops into a unique shape with distinctive surface patterns. Okay, so it creates a functional diversity required for thousands of cellular activities, such as hormones, structural proteins, those sorts of things. Okay, now there's this term called the native state, and the native state is just the functional three-dimensional form of a protein. Okay, whatever it is shaped like. Okay, a protein can become denatured though, and what happens is if you break bonds because of things like high or low temperatures or pH change, what it does is it breaks bonds on these proteins, which changes the shape. So the example that's usually used to illustrate denaturation is cooking an egg. So an egg is mostly protein. If you change the bonding between these protein molecules, you end up turning it from a semi-liquid to a solid, like when you cook an egg, and you can't go back. Okay, so some examples of proteins. Enzymes. So enzymes we use in the body for catalysts. A catalyst is something that speeds up a reaction because we aren't able to, normally the way we speed up reactions is by heating things, okay, because it speeds up the molecules and then it increases the chances of them bonding with each other because they're moving faster. We can't do that in the body though, because we have to have a temperature range somewhere within a few degrees of 98.5, right? And so in order to speed up processes, we use these enzymes, and they create bonds at a much higher rate. We have these things called antibodies, okay? So antibodies are these proteins that float around in the blood that are created by our lymphocytes, B lymphocytes, that cause, or that are used to attach to foreign objects. And what they do is they trigger a response of white blood cells and this thing called complement to destroy pathogens. Okay, so that brings us then to nucleic acids. So an example of nucleic acid is DNA. So DNA is the code or the genetic instructions that we have for our heredity and most other organisms' heredity, right? RNA is very, very similar to DNA, okay? So they call these helper molecules responsible for carrying out the DNA's instructions. There's, like we said earlier in the chapter, several kinds of RNA. What we do with the RNA, one of the things is we translate the program from the DNA into a protein. We just make an RNA molecule based off of a DNA molecule. We then take that RNA molecule with the instructions on it out to a ribosome, and we use the bases, these triplet bases called codons, to place an amino acid onto a chain, and once we finish making that chain, we end up with a protein. Okay, so the DNA is made of a double helix. So you're looking at a double helix here. Okay, so you can see it's twisted up with these bridges in between. Now they've sort of changed the way it looks right here in the middle to illustrate what we're looking at, and untwisted it. So here's one side and here's the other side. So you got to think of this like a ladder, and these are the rungs of the ladder right here, these hydrogen bonds in between these bases. Okay, so it's two complementary strands, and what we mean by complementary is where we see an A, we see a T on the other side, and where we see a G, we see a C on the other side. So formed by two very long polynucleotide, meaning many nucleotides put together, linked by hydrogen bonds between the nitrogenous bases, okay? So here's how DNA bonds. Adenine always pairs with thymine, so A goes with T, and guanine always pairs with cytosine, so G always goes with C. So I remember this in my mind. When I think of the bases, I always think ATGC, and that's how I remember A goes with T and G goes with C. I don't remember AGCT, for example. That would confuse me. So you may want to kind of always think of that as ATGC. RNA is a little different, okay? So RNA does not exist as a double helix. It exists as a single strand, except in some viruses. It contains ribose as the sugar instead of deoxyribose. That's why RNA is called RNA and not DNA. DNA stands for deoxyribose, which, as you recall, is a pentose sugar. So it contains uracil everywhere where you would have seen a T in DNA. Instead of seeing a T in RNA, you see a U. So A bonds with U and G still bonds with C like in DNA. So when we're making a complementary strand of RNA off of a DNA molecule, if the DNA molecule was ATGC, the RNA molecule would be UACG, and that would match up with it. Okay, so there's three major types of RNA besides those short chain RNAs we talked about earlier. They are messenger RNA, which is little m and then RNA. Ribosomal RNA, which is rRNA, and transfer RNA, which is tRNA.
The structure of the DNA molecule was first inferred by James Watson and Francis Crick, based primarily on X-ray crystallography data collected by Maurice Wilkins and Rosalind Franklin, and chemical analysis of base composition of DNA conducted by Irwin Chargaff. The key features of the structure are its right-handed double helical structure. Each helix consists of an alternating sugar-phosphate backbone with nitrogen bases projecting toward the interior of each helix. One complete 360-degree turn of the helix covers 10 bases of length and equals 3.4 nanometers in physical distance along the axis of the molecule. The width of the double helix is 2 nanometers. The nucleotide bases are attached inside each backbone of the molecule so that the nucleotides in one helix or strand are hydrogen bonded to the bases in the other helix or strand. The hydrogen bonds hold the two strands of the double helix together. An adenine in one strand is always paired with a thymine in the other strand, and a guanine in one strand is always paired with a cytosine in the other strand. Note that guanine-cytosine base pairs form 3 hydrogen bonds, while adenine-thymine base pairs form 2 hydrogen bonds. This makes guanine-cytosine base pairs more stable than adenine-thymine base pairs. Nucleotide pairing between strands also allows the sequence in one strand to determine the sequence in the complementary strand. The two ends of a strand are not identical. Note that on one end of each strand, a 3' hydroxyl group of the deoxyribose sugar is not involved in the backbone, or we say it's free, while at the other end of the same strand, the 5' hydroxyl group of the deoxyribose sugar at the end is free or may contain a phosphate that is free and not bonded to another deoxyribose sugar. This dissimilarity of the two ends of a strand creates the ability to uniquely distinguish each end of the strand. Because of this polarity of each strand, we can see that the two strands of DNA are oriented in opposite directions, or we say that they are antiparallel. In three-dimensional space, the two helices are not equally spaced along the axis of the molecule, such that a major groove and a minor groove are created by the asymmetric spacing of the antiparallel helix backbones. The major groove and minor groove play important roles in the binding of proteins that regulate gene transcription.
Okay, so let's talk about ATP. It's another nucleic acid. We've talked about DNA and RNA, and so this is the third. So ATP is adenosine triphosphate. It's called triphosphate because it has 3 phosphate groups on it. So it's a nucleotide that has an adenine on it, a ribose, which is a pentose sugar, it's the one in RNA, and 3 phosphate groups, okay? So here's the pentose sugar, here's the adenine, and here is the 3 phosphates. And this shows the same thing here. There's the 3 phosphates, pentose sugar, adenine over here. So it gives off energy when the bond between the second and third phosphate is broken. So this is a decomposition reaction, and remember that when a chemical bond is broken, it releases energy, and that's the whole reason why we create ATPs, is so we can use them sort of like little batteries. So when we need to, turn on our flashlight, we throw in a battery and we use up the energy from it. So when we break the bond, it results in an ADP. So when we take off one of the phosphates, it changes it from being a triphosphate to a diphosphate. So we call it ADP at that point. Okay, so let's talk about cells. We talk about them all the time, and by now in your education you guys should all know what a cell is. But if you remember the levels of organization, chemical, cells, tissues, and so on, you remember that cells are made up of a whole bunch of chemicals that work together to form life. Okay, so they can be as a single cell, or they can be subunits of a multicellular organism like people, where you have different types of cells. So some fundamental characteristics. So cells tend to be spherical, polygonal, cubical, or cylindrical, meaning cube-shaped, cylinder-shaped, like a polygon, so a pentagon or something like that. depending on the type of cell they are, and that all has to do with the chemical bonding in their structure. So protoplasm. Protoplasm contains the cytoplasm and the organelles. That's the stuff inside the cell. It is encased in a cell membrane or a plasma membrane or a cytoplasmic membrane, different names for them. Cells have chromosomes that have DNA on them, and ribosomes in order to create proteins. Very complex in function, and there's two basic categories that we've already mentioned, eukaryotic and prokaryotic cells. So eukaryotic cells, generally large in comparison to the prokaryotes, as we said earlier, found in animals, plants, fungi, protozoa, and these are the ones that have the organelles in them that we use for all kinds of different things. And because they have organelles, we have compartmentalization, so each organelle is its own compartment. Prokaryotic cells, on the other hand, no nucleus and generally no other organelles, complex structure that engages in nearly every activity that eukaryotes can. So many can function in ways that eukaryotes cannot, and as we go through this book, we are going to discuss those different ways. Okay, so cell membranes are composed of what? Mainly nucleic acids, proteins, phospholipids, or carbohydrates, or steroids. And although there are carbohydrates and proteins in them, as well as steroids in the form of cholesterol, the main component of cell membrane is definitely phospholipids. Okay, naming, classifying, and identifying microbes. Okay, the whole science behind classifying organisms is called taxonomy. Started by a man named Linnaeus back in the 1700s, and he started to establish these categories called taxa. Singular for that is taxon. So what it involves is nomenclature, which is the naming of them, and the classification, meaning putting them in specific categories, as well as being able to identify them. If you can't identify them, you can't name them or classify them, of course. Okay, so the assignment of different scientific names to these categories or individual organisms is called nomenclature. It's a fancy word for naming, okay? So we use this thing called binomial nomenclature in science. And this is the scientific name of the organisms, and it's a combination of two things, the genus and the species. That's what binomial means. Bi means two, nome means name. Okay, so pertaining to two names. Okay, so scientific names are italicized when they are written in print. So, example, Escherichia coli is italicized because it's the scientific name for a certain type of bacteria. Now, some of these have common names. Okay, E. coli, to my knowledge, does not. We just call it E. coli, but when they do have a common name, the common name is not italicized. Okay, now when we write them by hand, it's very difficult to italicize when you're handwriting, right? So what we do instead is we underline them when they are written by hand. So when we abbreviate it, we abbreviate the genus name with just the first initial, okay, and a period, and it's always capitalized. Okay, then we write out the full species name like E. coli. Notice the coli is not capitalized. Okay, so a lot of these names are often descriptive. When they're deciding what to name something, they kind of think of some of the things about it, and they try to come up with a name that maybe reflects that. For example, Staphylococcus aureus comes from the word staphule, which means a bunch of grapes, because a Staph aureus looks like a bunch of grapes. Lacto means milk. And so Lactobacillus san Francisco is a bacillus that digests milk that was discovered by a scientist, I believe, somewhere in San Francisco, something to do with San Francisco. Okay, so the classification is organized into a hierarchy of descending ranks. So it begins with a general all-inclusive taxonomic category, and then it gets more and more specific as we move down until eventually you end up with a specific species or subspecies. Because here are the categories. We start with the domain. Then we subcategorize it into kingdoms, then phyla or divisions, and you can call them either one, that's just different names for them. Classes, orders, family, genus, and finally species, and we should probably also mention subspecies as well. Okay, so origin and evolution. So with the theory of evolution came new ways of categorizing based on relation among organisms. And this whole concept was called phylogeny. And so this picture here in your book just illustrates humans versus protozoa. So you could use any number of different organisms, but this just shows each one of these going from domain all the way down to species. So if you look over here, you can see that the domain, both of these are part of the eukarya domain. All right, they're eukaryotes. But humans belong to the kingdom Animalia, and protozoa belong to the kingdom Protista. Okay, the phylum Chordata, that's because all of these animals are chordates. They have what's called a notochord, which in humans becomes the spinal cord, or at least part of the spinal cord. Matter of fact, same with all mammals. The protozoa is broken down into Ciliophora. That's the phylum, because everything on here has cilia, which are little projections off the cell membrane. Mammalia, you can see that we lose this spongy thing, whatever this is, I don't even know. And what we're left with is only mammals, and they have mammary glands and hair. The class of protozoa is Hymenostomia, which you can read what they have, and you're sort of getting the idea, I would think, okay? Until eventually we just narrow it down more and more and more. You can see that apes and humans are both part of the same family, but we get rid of this King Julian here, until eventually we end up with the genus Homo and species Sapiens, and this shows the characteristics of those. And we end up with the same thing here, Caudatum, and the protozoa, which is what species this protozoa is. It's a type of a paramecium, so genus Paramecium. Okay, so there's this thing called the universal web of life. And it used to be the way we thought of phylogeny was this phylogenetic tree, where there was one organism that branched into two, which branched again, and so on and so forth. And the way they determined this was based on what the knowledge they had at the time. But what they're starting to discover is that maybe it's not quite that linear, and so they're starting to think of this in this concept of this web of life, which we'll get to a little bit later. But Darwin and this Haeckel guy proposed two kingdoms when they began to classify animals, and they were just plants and animals. Okay, but of course, people started to discover there were things that didn't fit into either of those two categories. And so Haeckel later on added Protista. So then we had three kingdoms. Okay? In the 1870s, he then added the kingdom Monera. And this person named Whitaker added fungi sometime later, to where we have the 5 kingdom system that we use today. And here they are: animals, plants, protists or Protista, Monera, and fungi. Now later on, a person named Carl Weiss and some person named Fox came up with another system. Okay, it's called the Weiss-Fox system, and I'll show you that in just a minute. But it's based on these ribosomal RNA sequences. And once they were able to look at things at a molecular level, what they discovered was that there was two very distinct differences in the molecular makeup of this RNA. And what they decided from that was that there must have been a separation early on that we didn't know about before. among the prokaryotes. And so because of that, they came up with two separate domains. So analysis of these sequence revealed a separate group for the archaebacteria called archaea. So we have domain bacteria, which they already knew about, but they had to add this archaea to the whole mix as well. Okay, so that's this concept of domains, which we didn't have before. Remember, we started with kingdoms before. So all living organisms belong to one of these three domains. And here they are: bacteria, archaea, or eukarya, which are the eukaryotes.
Living organisms are divided into three major groups: the bacteria, the archaea, and the eukarya. The establishment and organization of these groups, called domains, is based on comparisons of nucleotide sequences of ribosomal RNA. Bacteria and archaea are similar in cell structure and are referred to as prokaryotes. These two groups can be distinguished by a number of important characteristics. All higher organisms, protista, plants, fungi, and animals share a common cell structure and are referred to as eukaryotes. Eukaryotic cells contain a nuclear membrane, endoplasmic reticulum, and organelles such as mitochondria and chloroplasts, whereas prokaryotic cells do not. In bacteria and archaea, protein synthesis occurs on 70S ribosomes, made up of 30S and 50S subunits, whereas in eukaryotic cells, translation occurs on 80S ribosomes, made up of 40S and 60S subunits. S values are based on the size, shape, and weight of a particle, which accounts for why the sum of subunits of the ribosome does not equal the S value of the intact ribosome. Histone proteins associated with the DNA of eukaryotic cells are generally lacking in bacteria, but present in archaea. Archaea histones are similar in structure to eukaryotic histones. Small amounts of protein are associated with maintaining the organization of bacterial DNA. Most bacteria and archaea have a cell wall. The bacterial cell wall contains peptidoglycan as well as proteins, polysaccharides, and lipids. The archaeal cell wall contains no peptidoglycan, but does contain proteins, polysaccharides, and lipids. Cell walls are also present in eukaryotes such as plants, algae, and fungi, but are lacking in animal cells and protozoa. Plant cell walls contain cellulose and lignins, whereas fungal cell walls contain chitin. The cell membranes of bacteria and eukarya contain phospholipids consisting of esters of glycerol, whereas the cell membranes of all archaea contain ethers of glycerol.
Okay, so that video we just watched, there was a lot of extra information that we will talk about in subsequent chapters. Pay attention mainly to just what we are going over in this chapter. Okay, so you got a little bit of a preview, but don't stress about that as far as this first of this chapter's quiz goes. Okay, so what we're looking at here is the Weiss-Fox system of taxonomy. As the video said, and as I said earlier, we used to think that there was only two divisions, prokaryotes and eukaryotes. But because of the ability to sequence the nucleotides in the RNA, what they have been able to discover is that there was a major branching very, very early on, and this archaea prokaryotic branch actually came off of this eukaryotic branch. So at some point there was a nucleus, and then it somehow lost its nucleus, because we used to think that it branched here where we had no nucleus, and everything on this side all came from this branch. So anyway, because of this, it's changed how scientists view the phylogenetic tree. So as I said earlier, they used to think that the phylogenetic tree was a lot more linear, but they're now starting to think of it as more of a web, because they said 40 to 50% of the DNA is transferred horizontally and not linearly up through the tree, like from a trunk to the branches, because of things like bacterial vectoring and things like that. Okay, so one thing to note, viruses are not part of the phylogenetic classification. Like I said earlier, they don't even, it's not even agreed upon whether or not viruses are actually a living thing, okay? So they did not take part in any of that tree that we showed on the last slide. Okay, so which of the following are domains of the Weiss-Fox system of taxonomy? Protista, Eukarya, Archaea, Bacteria, Fungi. And it's Eukarya, Archaea, and Bacteria. Now remember, Protista and Fungi are further on down, but not at the very beginning of that Weiss-Fox, those three domains.